Good afternoon. It's a pleasure to be here. So it is well accepted that the concise application of clinical endpoints is critical in lupus research. Composite endpoints of improvement have been recently developed, such as the SLE Responder Index and the BILAG-based composite lupus assessment, with a purpose to reinforce the strengths and shore up the weaknesses of their individual component measures. These endpoints are also able to account for improvement and worsening at the same time in different organs, and overall are thought to provide better discrimination between treatment and placebo. Nonetheless, their comparative analysis of their performance have been hampered by the few studies in which both have been analyzed, as well as by issues related to data quality in some of the older studies. It should be mentioned that biomarkers, although are not universal, in certain subsets of patients, a change in characteristic biology could provide an objective standard against which to compare clinical endpoints. Let's briefly review the definitions of SRI and BICLA. An SRI response requires a 4 or more point improvement in Selena SLEDAI from baseline, as well as no worsening in BILAG, meaning no BILAG A or more than one BILAG B scores, and no worsening in PGA that is less than 0.3 or 10% increase from baseline. The BILAG-based composite lupus assessment requires, again, improvement here in BILAG, meaning that all baseline A and B scores should improve by 1 letter grade or more, along with no worsening in BILAG, meaning no new A or more than one new B scores, and no worsening in SLEDAI 2K. Along with that, there should be no worsening in PGA and no treatment failure, defined as any non-protocol treatment. It should be noted here that although the requirement for no changes in medications is not in the definition of the SRI, in the large trials that utilize the SRI today, and that is the BLISS studies, no medication changes were allowed during the latter months, likely accomplishing the same end. So, the clinical components of BICLA and SRI can be used in isolation of medication restrictions, and in this case, they simply determine improvement. If we add on the medication restriction rules, we can better assess the response to a specific therapeutic agent that was given at baseline without clouding that assessment by the impact of medication changes. In our study, we compared the BICLA and SRI definitions of improvement to a gold standard of physicians' clinical assessment, each with and without inclusion of medication restriction rules. So, we identified participants of the Oklahoma lupus cohort who met the 1997 SLE classification criteria, had 2 clinic visits with BILAG, SLEDAI, and PGA scored, and had a SLEDAI of 6 or more at the baseline visit. We reviewed their medical records, included their labs, and each individual was globally rated by the physician at the second visit as same, worse, or significantly improved compared to baseline, based on the overall impression of response. Medication changes were recorded between the baseline and follow-up visits, but these were not factored into the physician rating of improvement. We also measure cytokine levels on stored serum samples from baseline and follow-up visits by a multiplex bit-based immunoassay. So, 91 patients were included. There were 5 males, 47 were Caucasian, 22 were African American, 14 were American Indian, and there were few Asian and Hispanic. Their mean age was 41 years, and the mean interval between visits was 6.2 months. At baseline, mean PGA was 1.75, mean BILAG was 15.1, and mean SLEDAI was 10. 68 of the 91 patients were rated as significantly improved at follow-up by the physician, whereas 23 were rated as same or worse. We assess clinical improvement by SRI and BICLA without medication limitations, and this was compared to the physician rating of improvement, or PRI. So as you can see here in this slide, there were among 68 patients that improved by PRI, 58 also improved by SRI-4, which is the SRI, and 52 improved by BICLA, leading to a sensitivity of 85% and 76% respectively. Among 23 that did not improve by PRI, 17 did not improve by SRI, and 18 did not improve by BICLA, with a specificity of 74% and 78% respectively. We also captured responses by SRI-3 and SRI-5, and these are defined similar to SRI-4, with the exception that a minimum of 3 and 5-point improvement in SLEDAI is respectively required. So spur rank correlations to PRI range from 0.6 for SRI-3 to 0.49 for BICLA, and all of them were highly significant. Upon a closer look, all 9 patients who improved by BICLA but not by SRI failed to achieve a 4-point improvement in SLEDAI. Nonetheless, 7 were rated as significantly improved by PRI. Also, all 16 responders by SRI but not by BICLA failed to improve in every organ. However, 13 were rated as improved by PRI. We also captured in response by SRI and BICLA here with inclusion of medication limitations. Under medication limitations, we included all changes in medications after the baseline visit, with the exception of NSAIDs and topical steroids. Here, the number of responders dramatically decreased, which led to a significant drop in sensitivity, down to 36.7% and 35% for SRI and BICLA respectively, which is actually not unexpected if you consider that the physician did not take any medication rules into account when considering the PRI response. The number of non-responders reciprocally increased, leading to an increase in specificity up to 95.6%. So that is when the medication rules are factored in as response criteria, this significantly decreases the likelihood that BICLA and SRI will detect an improvement when the physician does not agree. Exploratory analysis in this case of serum cytokines allows some preliminary modeling. In the case of IL-6, this was detectable only in a minority of patients at baseline, only 38 out of 91 patients. As you can see here in this figure, among patients that responded by PRI, there was a significant decrease in serum IL-6 between baseline and follow-up, whereas no such trend was observed in non-responders. When response was defined by SRI and BICLA, a similar change in IL-6 between baseline and follow-up was observed in the responder group, whereas no significant difference was seen in the non-responders. To conclude, shortfalls of the SRI and BICLA are usually due to the BICLA requiring only partial improvement, but in all organs, versus the SRI requiring full improvement, but not necessarily in all organs. Medication restrictions decrease the sensitivity and increase the specificity of these endpoints. And the physician's overall opinion, as well as the composite endpoints of improvement, corresponded to improvement in IL-6 in a subset of our patients. Thank you.
This paper is now open for discussion.
Yes, I don't know why you choose IL-6 levels to follow responders or non-responders, but whatever that is, the decrease in IL-6 level in the responders probably is much more related to the fact that the inflammation that is decreased because they are responding to the treatment. It has nothing to do with lupus pathogenesis.
I didn't hear your whole point. The decrease is due to what?
if you had another cytokine that decreases the inflammation, probably the results will be the same. This has nothing to do with the pathogenesis of the disease. It's a reflection of the fact that by responding to treatment, they have less inflammation.
Yes, that is actually a good point. I'm not sure how best we can interpret that. This can certainly be an effect of treatment here, but certainly as they improve, there's a decrease in IL-6. We do have to correlate medication change in this patient. We haven't had that done yet.
Are you suggesting that in reality the only thing we need is for the doctors to say whether the patient is better or not, because that is performing as well as all the other measurements? Is that what you're saying?
If IL-6 is the... I
didn't say that. I said, what you are telling us is that physicians responder, just the overall gestalt.
Okay, yes.
It's as good as the SRI and the BICLA. Then, then we don't need anything for the clinical trials, just the gestalt of the doctor. Are we actually going backwards or forward?
Yes. I'm not really suggesting that. This is a small study. Actually, there was indeed good correlation between the change in SLEDAI, BILAG, PGA, and the number of BILAG domains with improvement in responders versus non-responders by PRI, the physician grading. I think it has to do a lot with the physician assessor, and it's certainly not something universal, and I don't think this is really the way we should go.
I have 2 questions.
Why did you choose 6 months between evaluations? Because I think it's a long time.
We did not really choose that. This was I think the mean. There was a variation between 1 month and 24 months. So we really picked up studies that we had available data and samples for biomarker analysis. That was our inclusion criteria.
I have 2 questions. Would you be able to calculate the specificity and sensitivity of PRI against BICLA and SRI? And would you suggest, if you find those results are better, that we go back to using PRI? My second comment is, these are all from one center. Do you think the physicians' training in BILAG and BICLA and SRI confounds these results?
Certainly, I can start from the second one. It does have a role. Here, in our case, in our cohort, there were just very few physicians that did these assessments. And probably it's not the best scenario to assess whether training has to do with the results. Regarding the first part, we did not really look into that.